This is the video for Lab 13, Miscellaneous Reactions. When identifying unknown bacteria, the first thing you do is a gram stain, and this helps you identify the shape of the bacteria and the gram reaction. In doing so, this allows you to eliminate different options and focus in on one set of potential identities. To definitively identify the bacteria, you need to generate a biochemical profile or an enzyme profile. So you need to determine which enzymes the bacteria has and what types of biochemical reactions it can facilitate. And of course, because we can't see those enzymes or the products of the reactions, we need to use indicators, which indicate that a reaction went forward. In previous labs, we have looked at carbohydrate metabolism and also in nitrogen metabolism, which involves proteins and amino acids. In this lab, we are going to look at miscellaneous reactions so they don't fall into a particular group. Catalase test would have been the first test in this lab. If you remember when I talked about oxygen requirements, I told you that when you mix gaseous oxygen with water, that generates oxides. And oxides, such as hydrogen peroxide, are dangerous to cells because they can oxidize DNA and proteins. So the result of that can be the death of the cell. Hydrogen peroxide is one of those oxides. And catalase is an enzyme that takes that hydrogen peroxide and breaks it down back into water and oxygen gas. So catalase is one of those enzymes that protects microbes from the negative effects of oxygen. For this test, you would have taken a colony of bacteria, placed it on one of the slides, and added a drop of hydrogen peroxide. So you would have taken a colony, you can see a little bit of yellow in the center of the colony, and added a drop of hydrogen peroxide. And if bubbles form, that is the oxygen gas. So that means that the catalase is present. It's breaking down the hydrogen peroxide into water and oxygen gas. So this is the example of a positive reaction. A negative reaction, you would have added a drop of hydrogen peroxide and no bubbles means that that hydrogen peroxide is not being broken down by catalase. So this would be catalase negative. Oxidase test would have been the second test that you would have performed, and the oxidase test is looking for the presence of an enzyme called cytochrome C oxidase. And this is an enzyme that is involved in the electron transport chain, and it is actually the last enzyme involved in the electron transport chain, and its job is to take that electron that has passed through those redox proteins and bind it to the final electron acceptor. So that's what cytochrome C oxidase does. To perform this test, you would have taken a filter paper disc, put a colony of the bacteria on the disc, and added a drop of oxidase reagent. If the disc turned purple, that would be a positive. If the disc remained with the color of the bacteria, that is a negative. So in class, we would have used filter paper discs, but it is also possible to use Q-tips or swabs, and again, purple means positive and no color or just the bacterial color is a negative. The next test would have been the DNase test, and this is testing for an enzyme that can break down DNA. Some bacteria have the ability to make this enzyme and then secrete it out into the environment where it can break down DNA in the environment. To test for the presence of DNase, you would have used a special type of differential media. Remember, differential media contains substances that change color depending on the type of bacteria that are grown on it. For this is an example of DNase media. It is regular TSA media that contains DNA and an indicator such as methylene blue, which binds to the DNA. When you perform this test, you would have divided the plate in half, and you would have tested 2 bacteria, 1 on each side, and you would have just inoculated a round area of bacteria, the 2 different types of bacteria on each side. If the bacteria is able to make DNase and secrete it out, then the DNA around the bacteria would have been broken down, and that would get rid of the dye, the methylene blue around it, because methylene blue only binds to DNA. So this bacteria is positive because you see that halo around the bacterial growth, meaning that the DNA has been digested. This bacteria does not have a clear halo around it, so this is negative for DNase. The next test would have been Esculin Hydrolysis, and this is looking at the bacteria's ability to break down the esculin. For this, the media you would have used is called bile esculin agar, and it is selective and differential. It is selective because it contains bile salts, which will inhibit the growth of certain types of bacteria, and it is differential in order to identify the ability to hydrolyze esculin. So for this, the original color of the media is this brown color. To inoculate, you would have stabbed down into the media with the inoculating needle, and if the media turns black, then that is a positive for esculin hydrolysis. If it remains that brownish color, then that is a negative result. The last test in this lab would have been for motility, and motility is looking for the presence of flagella. The media you would have used for this lab would have come in a test tube, and it's less dense than normal agar. To inoculate this media, you would have stabbed down the center with the inoculating needle, and because the media is less dense, if bacteria have flagella, they would be able to grow away from the stab line. If they do not have flagella, their growth would have been restricted towards the stab line. And this media also contains an indicator which turns all bacteria red to make it easier to see the bacteria. This is an example of a non-motile bacteria. So as you can see, all the growth is concentrated around the stab line. And this is an example of a positive test. So the bacteria, the stab line was probably in the middle, and the bacteria were able to move away. So you'll see sort of fuzzy generalized growth. Some bacteria are not this motile, and what you would see here is just sort of general fuzzy growth moving away from a stab line. So this is also a positive, and it looks more blurry or fuzzy compared to that one. If your bacteria is a coccus, it must be non-motile, because remember, only bacilli and spiral bacteria have flagella.